It's a pleasure for me to introduce today's first plenary speaker, who is Tim Lenton. I just met Tim yesterday. He's a professor at the University of Exeter in the United Kingdom, and he's the chair in climate change and Earth system science. Tim's really known for his work on, well, for a number of things, but he's done a lot of work on the tipping points in the climate system and written extensively on that topic. He's also written an important book, a general book called Revolutions That Made the Earth, where he traces big changes on the planet from the past, and I think thinks about a little bit about the future. He's really, I was trying to think, you know, is he a geologist? Is he an oceanographer? He's in a geography department and teaches classes on physical geography. He's none of those things, or he's all of those things somehow. And he really thinks about, I think he really thinks fundamentally about how life has changed the planet and how the planet perhaps changes life. And we're going to see some of that in his talk today. So Tim, please come up and give us your thoughts.
Thank you, Mike. That was a lovely introduction. It's a real honor to have a plenary talk here. So I'm going to talk about some work we've been doing as a group. It's sort of an Earth system scientist's attempt to understand how phytoplankton work and their role in the marine ecosystem, and try and take some kind of new modeling approach to that would include acclimation and adaptation of the organisms to try and get a better understanding of how they might respond in a changing world. So this is work with my group. It started with Hal Williams, then Jim Clark came on board as a PhD student and later a postdoc, and then Stuart Daines became kind of in many ways the architect of the model. And I want to thank these other colleagues from my old university, University of East Anglia, molecular biologists. I'll talk a bit about our work with them. But first of all, the motivating questions. As a modeler, I would love to have a model that helps me understand something about the co-evolution of life and the planet, knowing that the marine microbial biosphere is the original biosphere, if you like, the original life forms on our planet. They've, in a sense, created the oxygen-rich world we're enjoying as oxygen-breathing animals today. They've lived through much more anoxic and sometimes sulfidic, or we call it ferruginous, iron-rich oceans, and as I said, ultimately created the oxygen-rich ocean that they're living in now. They've experienced all kinds of mass extinction events along with the rest of the biosphere during the Phanerozoic. So I'd like a model that can get some handle on how the whole evolutionary lineage of the phytoplankton has emerged, but I'd also like a model that can give me some handle on how the marine biosphere, that I know is incredibly rapid in its capacity to acclimate and adapt, especially down at the microbial level, how it might respond to the multiple stresses and forcings of a changing planet that our species is busy creating. And finally, thinking about these things kind of forced me and the group into trying to think through this eternal 64,000 euro question, really, of how do you cross the scales from the very smallest scales of biology and biochemistry, the gene scale, all the way up to what I will call briefly Gaia, the Earth system, going through the stuff that many of you in the room care about, our understanding of ecosystems and biogeochemical cycles. So this talk is really about our attempt to make that bridge. So I'll say a little bit about the model we're developing, known as EVE for short, the Evolutionary Ecosystem Model, then get on to some real science. I want to point the model at a problem I've been fond of for a long time: what determines the Redfield ratio, the N to P composition of the phytoplankton. And we'll have a look at some results, hopefully, where the model produces, I will call them emergent growth strategies of phytoplankton and corresponding predictions of their N to P composition. And we'll have a little think at the end about how that might change in a changing world and how we can make the bridge to genomic data. So traditionally, how do we approach modeling the marine ecosystem? Some of you will know this, but remarkably, we're still, at least in global climate models, running fairly crude representations of the marine ecosystem based on kind of 1940s models of nutrient uptake and growth, or a simple NP-ZD nutrient, phytoplankton, zooplankton, detritus models of a very crude food chain, if you like. There's reasons why this is the case that maybe I won't dwell on, and certainly we now have much more apparently sophisticated models with many functional types performing different biogeochemical functions within a global ocean model. But they're still at some level tied back to this earlier parentage. And we know that if we tune those models locally to good observations, they're very good then locally, but they're not particularly portable to another situation. And they certainly don't have these key properties that the microbial biosphere has in abundance, which is rapid acclimation of individual cells to changing environmental conditions, and also the capacity to genetically adapt as populations pretty quickly relative to the timescales of even human-induced global change. So how could we come in with a different line of attack? It's not new in any sense, the fundamental idea. We're basically taking, I think, what you'd call an evolutionary ecology approach that's been popular in terrestrial ecosystem science since the 80s. And the model that we build on is actually emerges out of work by Brian Shooter in his PhD in the 1970s. I guess the fundamental idea is that let's assume that organisms have been optimized by evolution, by natural selection, and at any given point in time, they've got some energy or resource that they're going to allocate to different traits or different parts, if you like, of the organism. And they'll do that in a way that at least maintains their fitness, if not improves it, because they're evolved to be that way. So it becomes a case of building a model where, I suppose the key change here is we want to open up the black box of the organism, if you like, and start to resolve some of the key subcellular components of, in this case, the phytoplankton cell. And I'm going to concentrate on those components that Shooter suggested that we should resolve: the structure of the cell, the biosynthesis apparatus, the photosynthesis apparatus, and we'll also keep a handle on some other components like DNA, and I'll say quite a bit about ribosomal RNA and its part in the biosynthesis apparatus. But hopefully, if we make a model built on these principles, the idea will be that if we can get the trade-offs between different traits right, then, and we set it in a realistic representation of the environment, providing natural selection along with the interactions with other organisms, then hopefully something will pop out of the model, emerge, that looks familiar. So to do this, we've got to be able to identify our building blocks of the organism. But great progress, of course, is being made on that. We know something about the key components, in this case of a phytoplankton cell. We know something about their phylogeny, if you like, and the phylogeny of the organisms. And we want to take that on board here. In what I show today, we're going to have just essentially one functional type, if you like, of phytoplankton cell. But in our model, we'll have some multiple instances of it that vary in a master trait of cell size. And each of those cells will be doing the same, will have the same sort of trade-offs in its what we'll call cellular economics. So just to put this on a map in terms of complexity, we want to do something that opens up the organism, so it's a bit more sophisticated than some monod-type growth model, but we don't want to go all the whole hog to a systems biology model. So out there now, increasingly, are some very elaborate, actually steady-state models built from molecular genetics of all the sort of biochemistry of, here I think it's a prochlorococcus cyanobacterial cell. We want to strike a balance somewhere in between, where we begin to resolve key parts of the cell, but we want it to be computable and understandable, so we're not going to go into all of this gory detail. So following Shooter, we've kind of got our model where key components are the cell membrane and other parts of the structure. We've got some DNA in there. We've got the biosynthesis apparatus and the photosynthesis apparatus as two key components of the cell. And the cells in the model world are going to be, they've got to be, the costs and benefits of making those components have to be grounded in observations. And of course, then out of that, some trade-offs can emerge. So like many of you, we're signed up to the philosophy of thinking in terms of functional traits of organisms and the trade-offs between them. So in this simple model where we have a sort of master trait of the phytoplankton cell size, but we have other traits, if you like, which is, think of yourself as the phytoplankton cell at a given time set. Shall I allocate more resource to light harvesting apparatus or to biosynthesis apparatus? Or do I want to be trying to concentrate on taking up as many nutrients as I can? Essentially, if you're trying to maximize nutrient uptake per unit cell mass, you want to be as small as possible. But obviously, as you get smaller, you've got less room for other parts of the other components of the cell. So you start to lose space to even have photosynthesis apparatus. So there's an inevitable trade-off that emerges there. And that's just one example of a few of the trade-offs that is there in even such a simple model. So we have this model of the cell. It's resolving some of the key subcellular components, some of the trade-offs between them. The way we put this in a model at the moment is we would have roughly 1,000 individuals that may vary in, say, size, the master trait, and are in each grid cell of a global ocean general circulation model, a version of the MIT Ocean GCM. We could, I think, implement the model in a more simple way and just have a sort of single master cell in each grid cell doing this kind of trade-off, and we might well get similar results. But we were interested in capturing some of the diversity of microbial life that you all know is abundant in the ocean, and for that reason we quite like the individual-based approach. We're also interested in life histories of organisms and the role they may play in ecology, and that's another reason to have an individual-based model. So we've got organisms populating some trait space. We've got our environment, if you like. In this model, it's basically a phytoplankton model with a sort of parameterization of size-based predation. So no explicit representation, I'm afraid, Steve and the others, of the zooplankton or the top-down controls. But let's see what we can get, even with such a crude model. So there's some interaction between different organisms and environmental variables that are acting as the environmental selection, the natural selection on what are viable growth strategies for the organisms. What's the right trade-off? What's a good trade-off strategy for the phytoplankton? And so there's a selection process that's picking out viable strategies in the trait space. And then from that feed back some effects, of course, on the environment. We have a single nutrient model here, so it's all very simple biogeochemically, but you've got to start simple to build understanding. So we happen to have phosphorus nominally as our master nutrient. I hope that gives a kind of an overview of the modeling approach, or the key parts of it. Let's begin to look with this relatively simple proof of concept model at some of the things you get out. So this is just the distribution of the single nutrient phosphorus, phosphate in this case, in the surface ocean. Far from perfect, but it gets some of the features like the oligotrophic gyres with very low nutrient levels, see a bit of the upwelling regions, and see the much higher residual surface nutrient levels in the Southern Ocean, for example. And over here we've got, okay, we populated the model with cells of all sorts of sizes, doing this internal trade-off between different bits of the cell. Which cell sizes kind of dominate in different locations? Well, perhaps unsurprisingly, in the oligotrophic gyres, very nutrient-limited situation, natural selection here is saying be as small as you can to be as efficient as you can at uptaking the very scarce nutrient resource. So we get a minimum cell size that's really tiny and is pushing the limits that are observed, and there's not much room left in the cell for other things. But if we go to the more nutrient-replete Southern Ocean, then here the minimum cell sizes that are popping out are considerably larger in this more nutrient-rich environment. Perhaps more interesting then, same model run, let's have a look at within the average cell, if you like, in each grid cell of the ocean, what is the allocation to cell structure, to light harvesting, to biosynthesis, so just as fractions of the cell. You see that the light harvesting doesn't have so much variation, but in the case of structure and biosynthesis, there's a lot of difference. So in those oligotrophic gyres, a great big fraction of the cell is its structure, because the cells are so tiny and you have to have some space for their DNA, which is explicit in the model, and some light harvesting, otherwise they're going to die. But they're really dominated by their structure. Go to the high latitude Southern Ocean, the more nutrient-rich environment, and suddenly where higher growth rates can be maintained, they can only be maintained because a lot of the cell is taken up by the biosynthesis apparatus, the manufacture of all the other parts of the cell. So that's the kind of generic model. Let me turn then to a specific science problem, especially for those for whom this is, whoa, that wasn't clever. Help. What did I do there? Anybody? Oof, thank God for that. Okay, apologies. So I don't know why Redfield sent the screen black. A great scientist, and as we all know, the Redfield ratio describes the average composition of marine organic matter with this canonical value of an N to P ratio of 16. But as we've known for many decades, that's the average, but there can be great variation in the composition of the phytoplankton and the other organisms in the ocean. And using this modeling approach, I was really interested to look at whether we could get an understanding of that variation in the N to P ratio of the phytoplankton. So some basic things to think about here are, well, in terms of key components of the organism, proteins, which include all your nutrient uptake or resource uptake apparatus and your photosynthetic apparatus, pretty scarce in phosphorus. The ribosomes, the ribosomal RNA that is key part of the biosynthesis of making everything else in the cell, is of course very phosphorus rich relatively, so N to P about 7:1. The membrane's relatively nitrogen rich and phosphorus poor, around an N to P of 22:1. And out of some mix of those key components comes on average N to P of 16. But of course we've been busy collectively reminding ourselves in the last 10 years that there's huge variations in these N to P ratios. So there's variation across different lineages of phytoplankton. Interesting that the older lineages, cyanobacteria and green algal lineages, tend to have systematically higher N to P, and somewhat younger red algal lineages tend to have lower N to P. If we try to estimate N to P varying spatially in the ocean, in this case from some kind of mixture of satellite chlorophyll recovery and some sort of size class weighting algorithm, we get a hint that there's probably higher N to P ratios of the organisms in the oligotrophic gyres, maybe up over 20, and lower N to P ratios in the very nutrient-rich, well-mixed high latitudes. Let's look at some real data for that rather than an estimate. These particle export N to P values, it's a little bit tricky to read this, but basically what you see in this lovely data compilation is if you pick out oligotrophic gyres, you can see some locations where you get very high N to P values. Go to high latitudes like the Southern Ocean and you'll pick out N to P sometimes down around 8 or half the Redfield ratio. So huge spatial variation there. The question is going to be, can our modeling approach help us understand that? And what we were going to do with the model is test this well-known hypothesis for what's governing the variations in N to P. I'm looking down at Jim Elser in front of me here, one of the key proponents of this idea. So the growth rate hypothesis would be that if you want to maintain a high growth rate, you're going to have a lot of ribosomes, phosphorus-rich ribosomal RNA. That is the basis of biosynthesis, and that's going to drag down your N to P cellular ratio. So in fast growth conditions, we should see lower N to P ratios of the phytoplankton cells. At low growth rates, if nutrients or resources in general are scarce, nutrients or light, you're going to need a lot of nitrogen-rich protein to acquire those scarce resources, whether they're light or nutrients, and that's going to swing you up to a high N to P ratio in the phytoplankton cell. So yeah, simple prediction of the growth rate hypothesis, we should be seeing lower entropy organisms at higher growth rates and the converse at slower growth rates. So if we're going to make a half-decent model of this, there turns out to be one really crucial model parameter. And apologies for the most heinously complicated plot of the whole talk, but this isn't a model plot, this is data. This is a data compilation of a lot of studies from many of them from the 1980s when it was still thankfully fashionable to take these really vital physiological measurements. And what we're plotting is RNA over protein by mass going upwards there. We've got a sort of temperature normalized growth rate of the photoautotrophs. This is all a big compilation of photoautotroph data. Entropy ratios are increasing downwards here, just to confuse things. And what we're trying to get at, we'd like to get at, is we would like to know how much of the phosphorus-rich ribosomes do you need to make the nitrogen-rich proteins, and how does that rate constant, what is that rate constant, and in a bit later we'll come back to how it might depend on temperature. But the central parameter here in a model is there's some rate constant, which is in a number of amino acids per ribosome per unit of time, here seconds. What should that number be? What should we prescribe it as in the model? It sort of represents a gradient in this space here. And there's a complicated compilation of data from all sorts of studies, all photoautotrophs. What is going on in this cluster of colored lines? Each color or each different symbol is a different specific organism tied to a particular study. Well, if we look at low growth rates on the left-hand side of the x-axis, all kinds of stuff is going on. You've got lines moving just vertically up or down the plot, rather suggesting that there are all sorts of ecological or evolutionary ecological growth strategies going on at low growth rates that are different from the growth rate hypothesis. We might discuss at length after the talk what they are. Let's focus a bit more on the higher growth rates where we start to see some lines sort of moving diagonally up the plot. We just made an estimate of a sort of a value for this key rate constant based on averaging 3 of the different data points and carrying some gray uncertainty range in what we think this crucial catalytic rate constant is. If we project it forwards up to sort of maximum conceivable growth rates, I'm not sure if you can see the numbers on the lines here, but they're N to P ratios you would get. And we would say even at maximum growth rates with this calibration, we wouldn't expect N to P to drop below about 12 there. We might get some, then we can bring into the play in a minute some other phosphorus contributions to the cell, but this is setting some sort of constraints on how much you can change the N to P ratio. So that was a little bit complicated, apologies, but hopefully you got the flavor. We've backed out of some exhaustive data compilation a number for a key rate constant. This is, I'll reiterate perhaps, this is how many amino acids, nitrogen-rich amino acids can you make per phosphorus-rich ribosome per unit time. Let's take our best guess on that number over here. Let's also make some assumptions about the phospholipid contribution to the cell. So the phosphorus in phospholipids is assumed to be 10% of the dry weight of the cells, and the phosphorus in DNA, 1% of the dry weight of the cells. And with those assumptions, we then get this prediction of how the phytoplankton N to P should vary spatially in the global ocean. And happily, it has some features that we've seen in the data compilations that exist. So we do predict higher N to P in the nutrient-poor gyres, and much lower N to P in the well-mixed high-latitude oceans. I can plot that. It always looks prettier on a map, but let me plot it. Let me collapse at least one of the horizontal spatial dimensions. Just plot predicted N to P as a function of latitude. And that's our sort of black line with the grey uncertainty bar around it, plotting the organism entropy on the Y scale there, and compare it to the compilation we were looking at earlier, the actual data for entropy. A little bit hard to do this, there's some funny change in the scale here. So I've plotted the sort of red stars taken over from the Martinet et al. Dataset onto our predictions, and I've also plotted this other partially model-derived prediction of variations in entropy from Weber and Deutsch. It depends whether you're a glass full or a glass glass half full or a glass half empty person, how you take this. But based on the growth rate hypothesis, we're certainly predicting some of the observed latitudinal variation in entropy ratios of the organisms, but we're not getting all of it. So what's going on? We get these very high entropy ratios in the oligotrophic gyres where nutrients are scarce because the cells are allocating resources to resource acquisition, in this case nutrient uptake machinery, that's proteins, that's very nitrogen rich. Their growth rate is low anyway, so they have a low amount of phosphorus rich biosynthesis machinery and ribosomes. As we move to these resource-rich, or at least nutrient-rich high-latitude regions, they can maintain a much higher growth rate. To do that, they have to put a lot more of the cellular resource into the phosphorus-rich ribosomes that are the engines of growth, if you like, and they have less of the proteins, and hence they have a lower entropy ratio. All making some sense, I hope. Clearly, there are some other ecological strategies at play, though. So if we can't get spectacularly high entropy values that are seen sometimes in the gyres, that suggests there's other ecological strategies. For example, that the structural phospholipid is substituted with something else. And we can't get the lowest entropy values in the high latitudes. That suggests that, for example, there are storage strategies for phosphorus that organisms living in those environments deploy. I'm going to look at Mike for a time signal. Excellent. Final bit of the talk then. What I haven't told you about is, so that's the first order control on what's varying phytoplankton entropy in this model world. But there is also, interestingly, a direct temperature effect. And this is where the link to molecular biology comes in. The ribosomes, like many other catalysts, if you like, have some temperature sensitivity to their function. And in essence, they are more efficient, catalytically efficient at higher temperature. So you need less, as a phytoplankton cell, you need less phosphorus-rich ribosome when you're living in a warmer temperature in order to make the same amount of every other bit of the cell, including all the nitrogen-rich proteins. So as things get warmer, to first order, your entropy ratio should go up because you don't need as many ribosomes. But I didn't get that sort of from a textbook. I got that from collaborating with some molecular biologists who were busy kind of proving this in their lab at the University of East Anglia. So it might look a little bit complicated, but we have, for example, some, especially to us non-molecular geneticists, but we have some Western blots here of some ribosomal protein just to show that a lot more of it's expressed at lower temperature. If we just looked at, well, there are two curves on this diagram over here. If we just look at the gradient of the curves over here, which have got time on the x-axis, but they're essentially a measure, the gradient slope is a measure of efficiency of translation or functioning of the ribosomes, and it's basically they're working more efficient at the higher temperature. Sorry if you can't see the details. And then Andrew Toseland, in his PhD, he looked at real field molecular genetic data across 5 different locations. They're ringed here. They're the Antarctic Ocean, Equatorial Pacific, North Pacific, North Atlantic and Arctic. And this kind of colorful plot is all about what variation can you see in transcription and expression across these environments. And the one thing that really stood out from this field data was, which you probably can't read, is translation here, is the thing that varies profoundly across these different temperature environments. And just to hammer that home, these are the terms enriched in the gene ontology, as it's called, in the cold environment, the coldest environment in the Antarctic compared to the warmest, the Equatorial Pacific. And out of that leaps the ribosome, structural constituent of ribosome translation, etc. So just hammering home that cold conditions, there's a lot more ribosomes needed than in the warm conditions to make the same amount of the rest of the cell. So that's one second order factor that's contributing to this gradient in entropy that we think is real in the real ocean and is popping out of our model. And it's not hard from that to infer that if you went into a warmer world, the pure physiological effect of a warmer world is that you're going to be more efficient at making proteins and you'll need less of these phosphorus-rich ribosomes. So on average, all the phytoplankton in a warmer condition should go to a higher N/P ratio. And it'll be fascinating perhaps to discuss after the talk, for those of you who are interested, what implications that could have for biogeochemical cycling, etc. But let me wrap it up, because I think my time's up. I hope I've shown you something interesting in this, I think, somewhat new evolutionary ecosystem modeling approach, guided by these fundamental questions of what are the functional traits underlying ecosystem structure, and can we understand those traits and their cost benefits in terms of something we can actually measure in a lab with our biological colleagues in terms of cellular physiology and biochemistry. Although it may look more complicated to start to resolve subcellular components in a global model, I'm kind of arguing that it isn't, that it enables us to represent some biodiversity and microevolutionary processes. It can actually reduce the number of free parameters in your model, and most importantly, link them to more measurable quantities, which is key. And it helps make this bridge to molecular biological data, which is proliferating hugely at the moment, and helps us also, gives us a framework to also start thinking about the macroevolution of the phytoplankton. And if we point that kind of approach at a particular problem, well, we can say that the growth rate hypothesis can explain some of the key N/P variation of the phytoplankton that we see in the ocean and in the lab. But of course, other evolutionary ecological strategies are also at play in the global ocean. Thanks for your attention.